The overall goal of this procedure is to quantify the level of neutrophil migration across a bladder epithelial barrier during bacterial infection or in response to chemoattractant molecules. This is accomplished by first growing cultured bladder epithelial cells to confluence on permeable supports. The second step is to infect the bladder epithelial cells with various bacteria or apply chemoattractant molecules to the lower reservoir of the permeable supports. Next, neutrophils are isolated from venous blood and are applied to the upper reservoir of the permeable supports. The final step is to enumerate the number of neutrophils in the lower reservoir after 1 hour using a hemocytometer. Ultimately, the transuroepithelial neutrophil migration assay is used to assess differences in neutrophil migration elicited by various stimuli.
Though this method can provide insight into aspects of the innate immune response during urinary tract infection, it can also be used to investigate other diseases, including bacterial infections that occur at other mucosal surfaces.
Perform this procedure in a tissue culture hood using aseptic technique. Using sterile forceps, invert the permeable supports in a sterile 25 mm deep tissue culture dish. For optimal results, use 5637 bladder epithelial cells that have undergone fewer than 10 subcultures. Trypsinize a 75 square centimeter flask of 5637 cells at 95% confluence, according to the written